Today we are going to discuss about bilirubin metabolism. We have already started with the topic jaundice. But to study jaundice further, that is what are the types of jaundice, how it is different from each other. For that, we have to learn about bilirubin metabolism. What is bilirubin, like how it is formed, how it is metabolized, etc. And only we can study about jaundice. Okay, so today we are going to learn about the bilirubin metabolism. I am explaining in a more simpler way, more brief way. So let's start with the bilirubin metabolism. So to learn about bilirubin metabolism, we can divide the metabolism into four stages, so it will be more easier for you to learn. So we'll take each stage and study in detail, so it will be more easier for you to understand also. The four stages of bilirubin metabolism are: first we'll learn about the sources of bilirubin, like from where and all we are getting bilirubin in our body. Second is the transport of bilirubin. For the metabolism to take place, always there should be transport of the product from one place to another. Then only the metabolism will take place. So the second is the transport of bilirubin. The third one, the more important one, is the hepatic phase. Hepatic phase, where liver comes into play. The last one is the intestinal phase. So there are four stages when we learn about bilirubin metabolism. First one is the sources of bilirubin. Second one is transport of bilirubin. Third one, hepatic phase. And fourth one is intestinal phase. Now we can learn about the sources of bilirubin. What are the sources of bilirubin? The main and the major one is the red blood cell, that is RBC. We know the lifespan of RBC. What is the total lifespan? What is the lifespan of RBC? Yeah, it is 120 days. It is 120 days. So after that we call it as senile RBCs. What do you mean by senile? Senile means old. Old RBCs. They will become old. So we know that we have seen that in old persons their skin will be like wrinkled, it will be wrinkled and get easily bruised like that. So likewise our RBCs also gets ruptured. Its cell membrane gets ruptured as it gets old. When it becomes old, its cell membrane gets ruptured and there will be release of hemoglobin. So that is the main source of hemoglobin and main source of bilirubin. That is, when RBCs gets ruptured, there will be release of hemoglobin. Next is, after the hemoglobin is getting released, our reticuloendothelial system will comes into action. That is, our tissue macrophages will come in the site. What they'll do, they'll phagocytose the RBC. They'll phagocytose the hemoglobin. They have seen a new thing, so they'll try to destroy it or phagocytose it. So they'll phagocytose or they'll engulf the hemoglobin. Okay. So now where is the hemoglobin? Now the hemoglobin is inside the macrophages. Now the further things are going to take place in the macrophages. Next, what they're going to do is they'll break down the hemoglobin. Okay, macrophages will break down the hemoglobin. They'll break down the hemoglobin into heme and globin. Yeah. So, what is globin? Globin means it is the protein part of the hemoglobin. That is the basic constituents of globin is the amino acids. So now we don't have to discuss that. We'll come into globin part later. Now we'll discuss about the heme part. Okay. So heme will further gets divided into a free ion and a straight chain of 4 pyrrole nucleus. So heme is getting divided into two things. Okay, one is free iron, and the second thing is what is it? A straight chain of pyrrole nucleus. The the major raw material of bilirubin is this straight chain. It is from this straight chain that what is produced? Bilirubin is further produced. Okay, so you can see this flow chart. Okay, hemoglobin, it is getting converted into heme is getting converted into biliverdin. You have to note that it is biliverdin that is produced first and not bilirubin. The first product is biliverdin. Okay, then only after that only bilirubin is getting produced. So there are some enzymes that is helping in this process. Okay, there are two enzymes which is helping. So in this first stage, we have to learn about two enzymes which helps in the production of bilirubin. Which are they? For the conversion of heme to biliverdin, what is coming into action? It is the heme oxygenase which is coming into action. Okay, heme oxygenase is the enzyme needed for the conversion of heme into biliverdin. Got it? Yeah. Next is, for that process, oxygen and NADPH is used. Okay, oxygen and NADPH is used for this conversion of heme into biliverdin. Next, the biliverdin is getting converted into bilirubin. So for that, which enzyme is used? Biliverdin reductase. The biliverdin reductase is used for this stage. So for the production of bilirubin, there are basically two enzymes which is used, that is heme oxygenase and biliverdin reductase. Here the biliverdin is getting reduced to form bilirubin. Okay, thus we have got the product now. We have got the bilirubin. Next is what our body, we have to see what our body is going to do with this bilirubin. Okay, one thing more I have to say is, I told previously that the major source of bilirubin is RBC. Around 80 to 85 percentage of bilirubin is produced from the hemoglobin, but the rest 15 to 20 percentage, from where it is coming? It is coming from the non-heme containing pigments. There are some non-heme containing pigments in our body. That is, there are pigments which does not contain iron. Okay, that it is non-heme containing means the pigments which does not contain iron. Okay, so some examples, I'll give you some examples. They are myoglobin, cytochromes and catalases. Okay, so we are done with the first stage, that are the sources of bilirubin and the production of bilirubin. We also learned about the sources and how the bilirubin is produced. Okay, we learned about the two enzymes that is coming into action. Next is the transport of bilirubin. That is how it is getting transported. So where we stopped with the bilirubin, now the bilirubin is produced within the macrophages. So now it has nothing to do with the bilirubin. The macrophages has got nothing to do with the bilirubin. So what they'll do, they'll release this bilirubin into the plasma. Okay, they'll release the unconjugated bilirubin into the plasma. Sorry, I forgot to mention one thing. The bilirubin formed here in the macrophages is unconjugated. What do you mean by unconjugated? That is, it is not bind to anything. It is free bilirubin. Okay, so it is this free bilirubin that is getting released into the plasma. Okay, the macrophages will release the bilirubin into the plasma. So what is taking place in the plasma? In the plasma, the thing that is taking place is the bilirubin will combine with albumin. In the plasma, bilirubin will combine with the albumin. So bilirubin is such a thing that it does not prefer to stand alone. It always want to combine with someone. Okay, so when it got produced, it was free. When it got released into plasma, it doesn't want to stand alone, so it got combined with albumin. The next phase, it will in each stage it will combine with something. Okay, so in the plasma, it is getting combined with what? It is getting combined with the albumin. So we are done with the transport of albumin. Now, next is the hepatic phase, the most important phase. So, what do you mean by hepatic? Uh, what you will get strike when you hear the word hepatic? That means it is something related to liver. Yeah, of course. So now the thing is we have reached till plasma. Okay, in the plasma it got combined with the albumin. Now this albumin bound bilirubin will slowly move on to the liver or to the hepatocytes. Hepatocytes means the cells of the liver. Okay. So in the liver, so many process will takes place with the bilirubin. So the hepatic cell membrane will absorb the bilirubin along with the albumin. Okay. So the thus hepatic phase starts there. As we divided the metabolism into 4 stages, we can divide the hepatic phase also into 3 stages. Okay, to understand it easily, we can divide it into 3 stages. First one is hepatic uptake, second one is conjugation, third one is secretion to bile. Okay, so this thing, like, we'll discuss the first one, hepatic uptake. I told you earlier that so many processes will take place inside the liver. Okay, in that, the first thing that is happening is what bilirubin will do. It will dissociate from albumin. It doesn't want albumin anymore. Okay, but it can't stand alone also. What it will do? It will get combined with a cytoplasmic protein called GST. What is the full form of GST? It is glutathione S transferase. So the first two things happening inside the hepatocyte is first dissociation from albumin and second it combines with the GST. GST means glutathione S transferase. Okay. Next what will happen? Next we are done with the hepatic uptake stage. Next we are going to discuss about the conjugation. What do you mean by conjugation? What is the meaning of the word conjugation? That is, it is getting combined with something. Okay. So there should be a reason for the bilirubin to get combined with the getting combined. There should be a reason. What is the reason? That is, unconjugated bilirubin is not water soluble, but it is alcohol soluble, but it is not water soluble. For the metabolism to take place, it should move further. It should move from the liver into the bile canaliculi, from bile canaliculi to the bile, from there to the intestine, from there to the kidneys. It have to move to so many places. But unconjugated bilirubin cannot move likewise because it is not water soluble. It is just alcohol soluble. Okay. So to make it water soluble, what it will do? It will get combined with the glucuronic acid. Okay. So that process is called as conjugation. Its combination with the glucuronic acid is called as conjugation. It takes place inside the endoplasmic reticulum of the liver, of the hepatocyte. Okay, ER, endoplasmic reticulum. Conjugation takes place inside the endoplasmic reticulum. So for that also some enzymes are needed. Okay, what is the name of the enzyme that is used for conjugation? It is UDP. You can see it in this presentation. It is UDP glucuronosyl transferase by the enzyme bilirubin UDP glucuronosyl transferase. Okay. Via this enzyme, bilirubin gets conjugated with the glucuronic acid. Two products are formed, that is, we can call it as bilirubin monoglucuronide or bilirubin diglucuronide. Okay, these things are formed inside the liver. These things are formed. Bilirubin conjugation is done. So, conjugation process is over. So, we are done with the hepatic uptake, conjugation, and next is secretion into bile. So, for the metabolism to take place further, it have to move from the liver into the bile canaliculi. Okay. So, to move into bile canaliculi, there is an active process. It is an active transport. That is, this conjugated bilirubin is getting actively transported to the bile canaliculi and is excreted to, and it is getting excreted to bile into the intestine. So it is where the bile, it is in the bile canaliculi that is bile is getting produced. It is so the conjugated bilirubin is getting actively transported into bile canaliculi. It is getting secreted into the bile and from there to the intestine. So that secretion into bile is also over. The transport here is active transport because energy is needed in this process. We are coming to the last and the final phase, that is the intestinal phase. So next is, so the conjugated bilirubin have reached the intestine. So what is next? For the conjugated bilirubin, it is present in the bile. The conjugated bilirubin is present in the bile. For it to get absorbed or reabsorbed or anything, it should not be absorbed. It cannot be absorbed as such. Okay. So here who will come into action? The intestinal bacteria will come into action. Okay. They will change the conjugated bilirubin into urobilinogen or stercobilinogen. It is actually urobilinogen and stercobilinogen. Okay. So in the intestine, conjugated bilirubin will get converted into urobilinogen and stercobilinogen. So there are three pathways. So to finish this metabolism, we'll give a heading as the fate of urobilinogen. That is, what is happening to urobilinogen. The first thing that is happening is some of the urobilinogen is getting absorbed by the kidney. Kidney absorbs it from the intestine and excrete urobilinogen through the urine. Okay. Second, the urobilinogen or the conjugated bilirubin will get converted into stercobilinogen, which is getting excreted through the feces. Okay. It is that which is giving that normal yellow colored stool. So the two pathways means fetus. One, it is excreted through urine. Second, it is excreted through feces. A small amount, what it will do, it is getting reabsorbed from the small intestine. It will go into its previous pathway. It will recycle. It kind of a recycling. That is, it goes into its previous pathway. It gets reabsorbed from the small intestine. Who will reabsorb it? It is the portal vein which reabsorbs the urobilinogen. From the portal vein, it reaches the liver, and from the liver, it gets again excreted into the bile. Okay. So when the portal vein comes into action, we'll tell it as enterohepatic circulation. What? Enterohepatic circulation. So we are done with the intestinal phase also. And another thing is, when the urobilinogen or stercobilinogen, when they gets exposed to air in the urine or feces, that is, urobilinogen is present in urobilinogen is present in urine and stercobilinogen in feces. When it gets exposed to air in urine and feces, it will gets converted into urobilin and stercobilin. So this is the end of the bilirubin metabolism. Okay. The next is This is a pictorial representation of bilirubin metabolism for you to easily understand. If you hear the explanation first and you look into this picture means you'll easily understand the bilirubin metabolism. As I already told, inside the macrophages, the hemoglobin gets divided into globin and heme. From the heme gets divided into iron and bilirubin. This bilirubin, it is the unconjugated one. Okay, that is the free bilirubin. Next is it gets entered into the intestine. Sorry, it gets entered into the hepatocytes where it combines with the first it will dissociate from albumin, then it combines with the glucuronic acid to form the conjugated bilirubin. Conjugated bilirubin. Then from there, via active transport, it gets transferred into the bile canaliculi, gets excreted into bile, and you can see it comes into the small intestine. From the intestine, there are three pathways. That is through kidney, it will get excreted as in the urine, and through intestine as continuation of intestine, it gets excreted in the feces as stercobilinogen. First in the intestine it gets converted into urobilinogen by the intestinal bacteria. Then it converts into, then it gets excreted in the urine and feces as urobilinogen and stercobilinogen, and the small amount is getting reabsorbed through portal vein back into the liver where it is getting re-excreted into the bile, and the process continues. Actually, it is a recycling. Okay, recycling is taking place of the conjugated bilirubin. That is, after it is getting converted into urobilinogen, it is again reabsorbed into the liver, and the process thus goes on. Next are some terms that I want to explain to you people. That is kernicterus. What is kernicterus? In newborn infants, sometimes we have to administer them some drugs like salicylates and sulfonamides. It will be due to some infections that we have to administer these drugs. But there are some side effects for these drugs. They are these salicylates and salicylates and sulfonamides. What they'll do, they will compete with bilirubin for albumin binding. That is, they won't allow the bilirubin gets to get binded with the albumin. Thus, it facilitates the bilirubin to enter into the brain in neonates and increase the risk of kernicterus. That is the jaundice of the brain. Okay. It will not allow the bilirubin to get combined with albumin, and thus bilirubin can easily enter into the brain of the neonates. This kernicterus forms. Next is bilirubin sulfate. Bilirubin sulfate, we told that in hepatocytes conjugation will takes place. Only 80%, only the major portion of the bilirubin gets conjugated with glucuronic acid. The rest 20% will combine with sulfate to form bilirubin sulfate. Next is ligandin. I already told earlier that in the hepatocytes bilirubin dissociates from albumin and gets combined with a cytoplasmic protein called GST, that is glutathione S transferase. Ligandin is another name of GST. Okay. Next is delta bilirubin. Delta bilirubin, it is the irreversible binding of conjugated bilirubin with albumin. That is irreversible binding. It cannot be excreted by kidney also. That is called as delta bilirubin or biliprotein. I'll give an explanation for that. Actually, conjugated bilirubin is bound to albumin in mainly two forms, that is reversible and irreversible. When actually it is reversible only, that is the binding with albumin is reversible only, but if it is present in serum for a long time, that is if any obstructive disease is happening, like bilirubin is not getting removed from the liver, that is the bile outflow is obstructed in some way, Due to some disease, the bile outflow is obstructed. Then what takes place? This reversible binding will change into irreversible binding. So, the conjugated bilirubin will irreversibly bind it to albumin. That binding is called delta bilirubin or biliprotein. And this bilirubin cannot be excreted by the kidney, and it will remain detectable in the serum for a long time even after the recovery from the disease, because since it is firmly bound to albumin, we can remove bilirubin if and only if it will gets dissociated from albumin, then it should combine with glucuronic acid, etc. So here it is irreversibly binding with what? Albumin. It cannot be reverted back, so it cannot be removed by kidney and etc. So it will remain in the serum for a long time. It is called as delta bilirubin or biliprotein. So we have finished with the bilirubin metabolism. Thank you.